unfold.
Welcome back to the Deep Dive. We are your guides, and we're here to take that dense source material, the scientific journals, the research, all that high-level analysis, and just pull out the essential insights, those aha moments you really need.
And today, wow, we are wading deep. I mean, right into the humid, spore-filled understory of the rainforest.
We are. We're tackling a subject that has literally jumped straight from mycology journals onto our screens all over the globe. The zombie ant fungus.
That absolutely fascinating subject. And its fame, in pop culture, especially with things like The Last of Us, that's no accident.
Not at all.
That fiction, it taps directly into two of our most deep, primal human fears, right? The fear of contagion and the terror of losing complete control of your own body, your own mind.
And you see these, frenzied, chaotic monsters in the stories.
Exactly. But the real organism, Ophiocordyceps unilateralis, it operates with this kind of surgical, ancient, evolutionary precision that is, I think, in so many ways far more compelling than any fictional monster.
That cultural hook is irresistible. It really is. It taps into something genuinely primal. But our mission for this deep dive is to move past the screaming aggression of the fictional infected.
Right.
We're going to spend the next 30-40 minutes meticulously deconstructing that fiction, but also reconstructing the dense biological fact of this organism. And I think most importantly, offering a sober, data-driven forecast about the actual threat of fungal disease in our warming world.
And as we go through the science, there are two core concepts for you, the listener, to just keep in the front of your mind. First, Ophiocordyceps is this perfect ancient example of what we call the extended phenotype.
Right. The idea that an organism's genes can influence things outside its own body.
Precisely. It manipulates the host behavior not for the ant's survival, not at all, but exclusively to maximize the fungus's own reproductive success. And second, and this is what separates this real-world biological marvel from the fictional apocalypse.
A big firewall.
The big one. It's what we call the thermal barrier. We have to dissect that concept today because it is the crucial defense mammals have against this entire kingdom of life.
Okay, let's unpack this. I think to really get the sophistication here, we have to start at the beginning, which is in the, often dry but fundamentally important world of taxonomy.
Yes.
Because the very name we use for this fungus is, a bit misleading.
Yeah.
In a scientific context, it's outdated.
Yeah.
For centuries, this thing was just sort of thrown into a massive bucket of similar-looking fungi, wasn't it?
That's absolutely right. We have to start with what mycologists call the Great Taxonomic Overhaul. Historically, the fungus was known simply as Cordyceps unilateralis.
Just Cordyceps.
Just Cordyceps. And it was classified within this huge genus, Cordyceps, which has well over 400 recognized species of entomopathogenic, so insect-infecting fungi.
Okay.
And up until the early 2000s, scientists relied really heavily on morphology.
Meaning just what it looked like.
Just what it looked like. They grouped species based purely on visible things. The shape of the fruit body, the stroma, the way the spores were arranged.
So if they had a similar-looking stalk erupting from an insect, they were just lumped together, even if deep down they were maybe distant cousins at best?
Precisely. And that approach worked, sort of, until the molecular revolution came along. Around 2007, a landmark phylogenetic study just changed everything.
DNA sequencing.
Advanced DNA sequencing. Researchers looked at multiple gene loci, these are like markers in the genetic code, to map out the true evolutionary relationships. And this study, it proved definitively that the traditional genus Cordyceps was, in fact, polyphyletic.
Okay, polyphyletic. That's a term we hear a lot in deep dives like this. Can you break that down? Why was that genetic distinction so critical here?
Sure. Polyphyletic means the group did not evolve from a single, recent, common ancestor. So think of it this way.
Yeah.
Imagine classifying all flying animals together. Bats, birds, and insects.
They all fly, but they're not related in that way.
Exactly. They all fly, but their common ancestor didn't fly. Their flight evolved independently, a process we call convergent evolution. That's what they found with Cordyceps. They had similar appearances because they evolved to fill a similar parasitic niche, but their genetic heritage was just too diverse to be a single cohesive genus.
So the family tree had to be completely redrawn, a massive reorganization. What did that end up meaning for our zombie fungus?
The consequence was a major taxonomic split. Scientists separated the traditional classification into several distinct families. Our star fungus here was moved to the family Ophiocordyceps and officially renamed Ophiocordyceps unilateralis.
Ophiocordyceps.
Ophiocordyceps. And this distinction is vital because it recognizes the genetic uniqueness of this zombie fungus lineage. If you compare the genera, the true cordyceps are often fleshy, brightly colored, sometimes used in traditional medicine.
Right.
Whereas Ophiocordyceps are typically tough, fibrous, darkly pigmented, and highly resilient. They're built differently, reflecting their specialization in manipulating ants.
And just when we get the new genus name down, we find out that even Ophiocordyceps unilateralis isn't one single species, but a whole complex of them.
Yes.
Which brings us to this idea of cryptic diversity, the sensulato complex.
Exactly. Scientists now refer to Ophiocordyceps unilateralis sensulato, which just means in the broad sense, because we're only just beginning to uncover the astonishing degree of cryptic diversity hidden within it.
And that diversity is all driven by one thing, right? Extreme host specificity.
Almost entirely. The leading hypothesis among mycologists is the one fungus, one ant theory.
One fungus, one ant. That implies this incredibly deep co-evolved relationship, that the fungus has fine-tuned its entire invasion strategy for a single species of host.
That's it, precisely. It suggests that for nearly every species of ant in the Campanotini tribe, that's the dominant group of carpenter ants this fungus targets, there may be a distinct co-evolved species of Ophiocordyceps that has specialized over millions of years to infect only that specific host.
So the chemical signals, the timing, the way it targets the muscles, it's all custom-tailored.
All of it. It's tailored. This hyperspecificity means the global diversity of the Ophiocordyceps complex is certainly vastly underestimated. We're only just scratching the surface when we call it unilateralis.
Do we have some concrete examples of this specialization from the field?
Oh, absolutely. We see this beautifully illustrated all over the globe. So if you go to the Atlantic rainforests of Brazil, researchers have identified Ophiocordyceps campanoti
Okay.
and it has evolved to target only the large forest and Camponotus rufapes. But if you travel across the globe to the tropical forests of Thailand, you find Ophiocordyceps campanoti leonardi, which has its own unique tailored strategies for its specific host, Camponotus leonardi.
And they can confirm this with genetics?
Yes, researchers confirm these splits by analyzing highly conserved genes, things like TEEF1, RPB1, and RPB2. They act like high-resolution genetic barcodes, allowing them to delineate unique species like O. fusiformispera or O. thanathanensis, each with its own specific set of adaptations.
That level of specialization, that kind of fine-tuning, that doesn't happen overnight. It must be the result of a biological arms race that stretches deep into geological time.
It does.
And that brings us to the really astonishing evidence of the death grip being 48 million years old.
This is truly one of the most compelling parts of this whole story. The partnership, or war, depending on your perspective, between this fungus and ants, is not some modern innovation.
Right.
The strongest evidence of its antiquity comes from the Messelpit fossil site in Germany. This site is famous for its just exquisitely preserved fossils from the Eocene epoch, roughly 48 million years ago.
So we're talking about an evolutionary mechanism that was perfected before the Andes Mountains even existed, before most modern mammals dominated the planet.
Essentially, yes. What researchers found at the Messelpit were fossilized leaves bearing these very distinctive scars on the major leaf veins.
And not just random damage.
Not random at all. They were dumbbell-shaped marks, morphologically identical in every single detail to the precise bite marks left by modern ants undergoing the terminal death grip of Ophiocordyceps.
And the fact that the bite specifically targets the vascular tissue, the vein of the leaf, that's the smoking gun, isn't it?
That is the smoking gun. It confirms the manipulation mechanism was fully established. The ant wasn't just chewing a random hole. It was forced to bite down with surgical precision on the strongest, most stable part of that leaf structure.
So the whole behavior was already there.
The whole behavioral manipulation, the forcing of the ant to climb, to navigate, to anchor itself, it was already fully established and perfectly executed 48 million years ago in that ancient paratropical environment.
That's just incredible. So what is that long history, this evolutionary stasis, what does that tell us?
Well, it supports a concept we call biogeographical parallelism. It means that even though the German Messelpit climate shifted dramatically over time, the coevolutionary strategy, that extended phenotype of the death grip, was so effective at ensuring the fungus's transmission that selection pressure hasn't forced it to change.
If it ain't broke, don't fix it. For 48 million years.
When a mechanism works perfectly for 48 million years, you know you are dealing with an evolutionary masterstroke. The perfection of this parasitic relationship predates much of the world as we know it.
So we have an ancient, Hyperspecific fungus. The question now is, how does it pull off the internal takeover? Once we move past the labels, we have to look at the life cycle. Section 2, the invasion plan. It starts with breaching the armor.
The ant is a fortress. It has this rigid, chitinous exoskeleton, its biological plate mail, and a formidable innate immune system. The infection begins not through ingestion, as you see with some parasites, but through contact.
Just by touching it.
Just by touching it. A foraging ant, walking on the forest floor, brushes against these microscopic ascospores that are raining down from a dead cadaver above, and those spores adhere to its cuticle.
So how does the fungus get through that armor? I mean, it can't just be brute force, can it?
No, it's a very sophisticated combination of mechanics and chemistry. The spore germinates, producing something called a germ tube that differentiates into a structure called the eprisorium.
The eprisorium.
And this structure acts like a biological drill bit. It concentrates pressure on a single point of the cuticle. But that pressure is only the delivery system for a highly specialized cocktail of hydrolytic enzymes.
Okay, so what are these enzymes doing?
Well, transcriptomic and proteomic analyses have identified the weapons. The fungus secretes chitinases, which are essential for breaking down chitin, the main structural component of the exoskeleton. It also deploys lipases, which degrade the waxy hydrophobic outer layer that gives the ant its water resistance, and various proteases that chew through the protein matrix. It's an enzymatic assault, effectively dissolving a tiny, precise hole right through the ant's armor.
And that lets the hyphae, the fungal threads, penetrate the body cavity, the 𝐏𝐃𝐂𝐃𝐂𝐏.
Correct. And once that hypha breaches the armor and enters the internal environment, the ant's equivalent of blood, it has to change form immediately.
Or else the ant's immune system would spot it and neutralize it instantly.
Instantly. This is the sophisticated stealth mode, the dimorphic transition into the blastospore phase.
A dimorphic transition.
Yes, this transition is a crucial survival strategy. Upon entering the 𝐏𝐃𝐂𝐃, the fungus switches abruptly from filamentous, thread-like hyphal growth to a unicellular, yeast-like phase, producing millions of free-floating hyphal bodies, or blastospores.
And what's the benefit of that switch? You said the blastospores have a dual purpose?
They do. First is dissemination. Being tiny and unicellular, they can circulate passively in the ant's hemolymph, the body fluid, like a systemic infection wreaking every organ, every muscle, every neurological node throughout the entire body.
And the second purpose?
The second and most critical is immune evasion. The ant's innate immune system, which is primarily composed of cells called hemocytes, is highly effective at recognizing and surrounding large foreign objects, especially long filamentous hyphae. They use processes like encapsulation and melanization to wall them off.
So the immune system is basically looking for long threads to attack. By becoming millions of tiny, isolated yeast cells, the fungus is just flying under the radar.
Exactly. The yeast-like form is far less immunogenic. The ant's hemocytes, they struggle to efficiently detect and engulf millions of tiny disconnected cells dispersed throughout the entire body cavity.
And this allows the fungal biomass to just grow and grow.
Exponentially. During the incubation period, which lasts anywhere from 9 to 14 days, all while the ant appears outwardly normal. This is the biotrophic phase. The fungus is feeding and building resources on the living host, conserving its energy for that single final manipulation event.
That is a masterclass in stealth. A slow, systemic buildup. But to execute the final command, the death grip, all those individual cells have to act in perfect unison. How do they transition back to form a coordinated structure? We talked about the systemic network.
Right. So as the terminal phase approaches, environmental cues inside the host, we think it's likely related to nutrient depletion or internal chemical signals, they trigger the differentiation of the hyphal bodies back into mycelia, or hyphae.
And they don't just grow randomly.
No, instead of just growing randomly, they form this dense, interconnected fungal architecture throughout the ant's entire body, penetrating and surrounding the organs and muscles.
And we have advanced imaging data on this, right? Which shows a physical mechanism for this coordination.
We do. Researchers use these highly advanced techniques, most notably serial block phase scanning electron microscopy, or SBSM.
Okay, what is that?
It's a technique where you embed the entire infected ant in resin, and then you use a diamond knife to repeatedly slice off incredibly thin layers, and you take a high-resolution image of each layer.
Thousands of images.
Thousands of images.
Yeah.
And then they're all reconstructed into a detailed 3D model of the internal fungal structure.
And what did that 3D reconstruction reveal about how this network actually functions?
The data show that the hyphae don't just exist side by side. They connect to one another via what are called anastomosing tubules. They literally fuse their cell walls to create channels linking cells across different parts of the ant's body.
So it becomes one single massive interconnected organism inside the ant.
A single, coordinated, body-wide biological network. And this network allows for the rapid transport of nutrients, signaling molecules, and critically chemical effectors. The fungus has essentially built a functional, parasitic secondary body, using the ant's exoskeleton and tissues as its scaffolding, all prepared to execute the final command.
The idea of a coordinated secondary body inside the ant that leads us directly to the core discovery that overturns decades of speculation. And it absolutely demolishes the fictional narrative.
It really does.
For years, the idea was that the fungus invaded the brain to hijack the central nervous system. But the science shows something far more fascinating.
It does. The 2017 study led by David Hughes and his team, using fluorescent microscopy to tag and track the fungal cells, that was the pivotal moment. For the first time, scientists had an unambiguous 3D map of the fungus inside the ant at the very moment of behavioral manipulation.
And what was the great revelation?
The revelation was that while fungal cells were densely abundant everywhere, in the legs, the thorax, the head capsule, they were conspicuously and consistently absent from inside the brain tissue itself.
So it wasn't in the brain?
Not in the brain. The ant's critical blood-brain barrier remained intact. The fungus clusters intensely around the brain, effectively isolating and besieging it. But there is no direct physical penetration of the neuronal clusters, specifically the mushroom bodies, which handle things like memory and complex decision-making.
So the ant's brain is still technically functional. It's still perceiving the world right up until the point of death. That suggests the ant is a prisoner in its own body, forced to execute commands it can't override.
That is the chilling implication. The fungus actually needs the ant's central nervous system to remain functional, at least until the final moment. Well, a truly brain-dead ant can't perform the high-level sensory processing required to navigate the forest understory, find the specific type of vegetation, climb exactly 25 centimeters, and execute the perfect bite on the leaf vein. The fungus preserves the CNS as a necessary piece of the machinery for navigation, but then it seizes control elsewhere.
And this is where it gets really interesting. If the fungus isn't pulling the strings from the central brain, then where is the control happening? This leads us to the revolutionary peripheral control hypothesis.
The control is localized. It's in the peripheral neuromuscular system, specifically the muscles of the mandibles and the legs.
The muscles themselves.
The high-resolution SPSM imaging confirmed that as the terminal phase approaches, the hyphae physically penetrate the individual muscle fibers. They wrap around the muscle cells and form what researchers suspect are essentially parasitic neuromuscular junctions.
So they can bypass the ant's own motor commands?
And stimulate the muscles directly, yes.
The death grip is the ultimate example of this. If the fungus is directly stimulating the muscles, that bite isn't voluntary at all.
It is absolutely not a voluntary action. It's a physiological tetanus induced by the fungal network in the mandibular muscles.
Tetanus, like lockjaw.
Exactly like lockjaw. When researchers examined the internal structure of these mandated muscles using electron microscopy, they observed extensive cellular destruction, damage to the sarcomeres, which are the fundamental contractile units of the muscle, and severe depletion of critical energy organelles like mitochondria and the sarcoplasmic reticulum.
So the fungus is essentially using chemical and mechanical damage to induce a state of rigor mortis, but just in the jaw and early.
Exactly. It induces a state of permanent tetanic lockjaw, specifically in the jaw muscles, while the rest of the ant is still physiologically functional enough to be alive.
And that ensures that even after the ant dies and the body relaxes, the mandibles stay clamped firmly onto that leaf vein.
It secures the fungal platform. It prevents the ant from falling back to the inhospitable ground. The ant's brain may perceive the world, but the fungus is driving the legs and locking the jaw at the muscular level.
But the manipulation isn't just physical penetration. I mean, if it's clustering around the brain and driving systemic behavior, it must be flooding the whole system with chemicals. What does the cutting-edge metabolomics profiling tell us about this so-called zombie cocktail?
The fungus wages an incredibly sophisticated chemical war. Metabolomic profiling of the fungal secretome, that's the mix of compounds it excretes, reveals this complex arsenal of neuroactive and cytotoxic metabolites.
And it's targeted.
What's truly astounding is that it appears to display targeted pharmacology. It upregulates different metabolites depending on the tissue environment. It has one set of chemical keys for when it's near the brain tissue and a completely different set for the distant muscle tissue.
Okay, let's delve into some of those specific compounds. How does the fungus chemically achieve the observed behavior, the climbing, the lethargy, and that final lockdown?
We can detail several metabolites that scientists hypothesize are key components of the manipulation. First, there's phingosine.
Phingosine?
It's a lipid signaling molecule found in high concentrations in infected ants. In mammals, it's involved in cell-to-cell communication. In insects, it's hypothesized to modulate the central nervous system, specifically targeting the ant's ability to coordinate complex movement.
So that could be what causes the drunken, uncoordinated walking you see before the climb.
Its presence strongly suggests it's responsible for inducing that characteristic lethargy, yes. Then second, we have guanidinobutyric acid, or GBA.
GBA.
This compound is a GABA analog. GABA is the primary inhibitory neurotransmitter in many nervous systems, but its derivatives can alter locomotion and behavior. The high levels of GBA suggest the fungus is actively altering the host's neurotransmitter balance, possibly inducing that final compulsion to leave the nest and climb, or maybe even inducing seizure-like activity to aid in the final bite.
So it's actively trying to confuse and disrupt the ant's normal social behaviors.
Absolutely. Then you have to consider hypoxanthine. This is a degradation product of nucleic acids, but it's found at high extracellular concentrations near the neural tissues. It's known to have deleterious effects on neurons.
So what's its role?
It potentially functions to damp the ant's high-level motor neuron function. It essentially acts as a chemical muffler, suppressing the ant's intrinsic instinct to override the fungal compulsion or, that necessary drive to return to the safety of the colony.
And you mentioned an antioxidant, ergothioneine. Now, why would a parasite secrete a compound that actually protects the host's tissues? That seems totally counterintuitive.
It is the ultimate parasitic control move. Ergothioneine is a powerful antioxidant. Its function is to protect the ant's tissues from premature decay or the oxidative stress that the infection itself generates.
Why would it do that?
Because the fungus needs the ant to survive long enough, to remain structurally sound and physiologically functional, to reach the precise target location and execute the death grip. The fungus curates the circumstances of death, ensuring the host is a perfect temporary vehicle, not a rotting corpse that falls apart too soon.
That is truly the definition of the extended phenotype. The fungus has evolved not just to kill, but to micromanage the life and death of its host to secure its own survival.
And that level of coordination required to achieve that perfect death scenario, the right place at the right time. That is the single strongest evidence of how deeply this parasite has integrated itself into the host's ecology and physiology.
It's not enough for the ant to simply die. It has to die exactly where the fungus can germinate and reproduce successfully, which leads us to the quantification of the kill zone.
Infected ants always exhibit what's called summit disease. They leave the relative safety of the subterranean colony and climb vegetation, but that climb is not random. It's programmed. Field studies conducted across tropical hotspots, notably in Thailand and Brazil, have quantified this kill zone with remarkable precision.
So what are the specific parameters? What defines this perfect fungal microclimate?
They are environmental hyperspecifics. First, the hyro, dead ants, are consistently clamped onto leaves approximately 25 centimeters above the forest floor.
Always 25 centimeter.
Consistently around that mark. Second, the surface. There's a near universal preference for the abaxial surface, that's the underside of the leaf. And third, the orientation. In many studies, researchers note a slight but statistically significant preference for leaves facing a north-northwest direction.
So it's even optimizing for the sun's path.
It suggests it's optimizing for specific temperature and light regimes, yes.
Why is that 25 centimeter underside location so absolutely critical for the fungus?
That positioning is the key to fungal success. It provides the optimal microclimate necessary for the fungal stroma to develop and release viable spores. That 25 centimeter zone is characterized by extremely high humidity, consistently between 94 and 95%, and a moderate temperature range, ideally 20 to 30 degrees Celsius.
And fungal spores are sensitive to drying out.
Extremely sensitive to desiccation and heat.
And what happens if the ant gets it wrong? Or if researchers intervene?
Well, scientists have done experiments where they take dead, infected ants that have successfully performed the death grip and they relocate them. If they move the cadaver higher into the canopy where it's too hot and dry, the fungus fails to produce a viable stroma. It just withers.
And if they put it on the ground?
If they move the ant down to the ground, it's either too wet or, more likely, it's too quickly scavenged by other organisms. The fungus sacrifices the ant's survival for its own reproductive fitness, and it demands this tiny, perfect bubble of microclimate. That specificity is a survival bottleneck.
That addresses the precision in space. But arguably the most intriguing field observation is the precision in time, the chronobiology of the solar-noon synchronization.
This temporal precision is truly mind-boggling. Long-term field observations have repeatedly shown that the final act, the descent from the canopy and the terminal desk grip, they don't occur randomly throughout the day.
When do they happen?
They happen almost exclusively around solar noon, the peak of the day.
How does the fungus synchronize itself to the Earth's rotation? Is it leveraging the ant's own internal clock?
The hypothesis is that the fungus is deeply integrated into the host's circadian rhythm, but it likely uses external cues to trigger the final event. The drivers are thought to be maximum light intensity, which would satisfy the fungus's innate phototropism, its desire to grow toward light and peak temperature.
Why use the hottest part of the day? That seems risky.
Well, by forcing the ant to bite exactly at solar noon, the fungus might be utilizing the ant's physiological response to peak heat and stress to accelerate that rigor mortis lockjaw mechanism.
Yeah.
The heat ensures rapid water loss from the mandibular muscles, which helps secure the permanent lock before the ant's cellular processes fully cease.
It's leveraging everything.
It's leveraging the host's internal biology and the external environment perfectly for its own reproductive timetable.
So once the ant is locked, the fungus has successfully used it as a sophisticated pre-positioned platform. The final stage is reproduction, leading to the stroma eruption and the formation of the infamous killing field.
Upon confirmation of death, the fungus executes another vital transition. It switches instantly from biotrophy feeding on live tissue to necrotrophy, rapidly consuming the remaining dead soft tissue.
And it has to protect its platform.
It does. It must also quickly colonize the entire internal structure and actually fortify the ant's exoskeleton to prevent it from decaying too quickly or falling apart before the stalk can emerge.
And that's when the visual horror really begins.
Over the next 4 to 10 days, the hyphae consolidate and force the growth of the stroma, the fruiting stalk, which emerges forcibly from the weakest structural point, typically the dorsal neck region.
And it's huge, right?
This stalk can be impressive. It grows several centimeters long, often reaching twice the length of the ant itself to maximize spore dispersal. And once the fertile head of the stroma matures, it releases infectious ascospores.
Which rain down.
They rain down, creating a concentrated, localized killing field in a radius of roughly 1 square meter beneath the leaf, ready to infect any susceptible ant foraging below and perpetuating the cycle.
We've established that this fungus is an evolutionary genius, executing its life cycle with just surgical precision. If it's this successful, the natural question is, why hasn't it wiped out the entire ant population?
Right.
And the answer lies in the ecosystem's fierce checks and balances, starting with the ant colony itself and this concept of social immunity.
Ant colonies are often described as superorganisms, where the survival of the group completely outweighs the survival of any one individual. Therefore, they have evolved these collective immune defenses, behavioral adaptations, employed by the entire colony to minimize disease transmission among nestmates.
And this is their primary defense.
This is their primary defense mechanism against persistent, pervasive threats like Ophiocordyceps.
So what are the specific behavioral defenses the ants employ to protect the colony?
The primary line of defense is allogrooming. Worker ants constantly and meticulously groom one another. And this behavior is crucial because it physically removes the spores from the cuticle before they have a chance to germinate and penetrate the armor.
It's like constant self-decontamination.
It is. Beyond that, there's extremely effective sanitation and removal. Healthy ants are biologically hardwired to detect sick or infected nestmates.
How do they detect them? You said the fungus is in stealth mode for weeks.
They detect them through subtle chemical shifts, likely changes in the infected ant's cuticular hydrocarbons. These hydrocarbons form the chemical scent profile that dictates identity and social recognition within the colony.
The colony's password.
Essentially. As the fungal biomass grows, it subtly changes this chemical profile. The moment an ant detects a shift indicating infection, often before the manipulation phase even begins, the infected individual is treated as a biohazard. They're often physically dragged far from the central nest or deposited in designated graveyards, sometimes miles away, to minimize risk to the central brood.
So the fungus forcing the ant to climb 25 centimeters outside the nest is a direct counter-adaptation to the colony's efficient chemical-driven corpse removal system.
It is the parasite's response to the host's defense. If the ant dies inside the nest, the fungus would be discovered, removed, and neutralized before it can ever produce a fruiting body. The kill zone strategy is an adaptation that ensures the ant dies in a location the colony cannot safely or easily police. It's a continuous biological chess match.
But the ants aren't the only forces fighting the zombie fungus. The most surprising ecological control mechanism is hyperparasitism, the existence of an anti-zombie fungus.
This is perhaps the ultimate check on the entire trophic cascade. Hyperparasitism refers to mycoparasitic fungi, so fungi that parasitize other fungi.
A fungus that eats a fungus.
Exactly. These secondary pathogens effectively attack and neutralize the reproductive capability of the zombie ant fungus, a phenomenon scientists sometimes call parasitic castration. Without them, Ophiocordyceps might indeed cause widespread collapse.
Can you give us some specific examples of these fungal heroes and how they operate?
We have several well-described examples. One is Polycephalomyces oviedoensis. This fungal species attacks the stalk of the Ophiocordyceps stroma, coating it entirely and effectively sterilizing it, preventing the release of its infectious ascospores. Another fascinating example is the newly described genus Niveomyces Ophiocordycipidus, which forms this distinctive white, snowy mold right over the Ophiocordyceps cadaver, neutralizing it before spore production can even occur. Simplicillium lanacinivium is another one noted in similar systems.
Does this hyperparasitism actually make a measurable difference in ant populations? How significant is this ecological check?
It is massively significant. Studies indicate that hyperparasitism is the most potent biological control agent in these ecosystems. Data shows that up to 55% of all Ophiocordyceps cadavers may be hyperparasitized.
Well, more than half.
More than half. In some field populations, researchers found that only a tiny fraction, maybe 6 or 7% of the fruiting bodies, successfully reach full maturity and release viable spores.
So the hyperparasite creates a dramatic bottleneck.
A huge bottleneck that ensures the stability of the ecosystem. It demonstrates the balance. The host, the ant, is regulated by the parasite, Ophiocordyceps, which in turn is fiercely regulated by the hyperparasite.
All right, we've established the incredible biological fact. The fungus is hyperspecific, ancient, and controls muscles, not the mind. Now we have to address the ultimate question, the one driven by the fiction. Is the jump to humans, the functional takeover of a mammal, biologically possible?
And when we rigorously evaluate the biological barriers, we find they are just immense. The single most important defense we possess, the one that prevents the vast majority of fungal species from ever being a threat, is the thermal barrier.
Mammalian endothermy.
Yes. This is the hypothesis championed by Dr. Arturo Casadevall of Johns Hopkins, which posits that our high body temperature is an evolutionary luxury we possess.
It costs a lot of energy to maintain.
Precisely. Dr. Casadevall and his colleagues argue that maintaining a body temperature of 37 degrees Celsius, or 98.6 Fahrenheit, is metabolically very expensive, but it pays off massively in immune defense. It filters out the vast majority of the fungal kingdom, estimated at well over 1.5 million species, which simply cannot survive or replicate efficiently at that temperature.
And we have specific thermal tolerance data for the tropical Ophiocordyceps. What is its absolute thermal limit?
Ophiocordyceps unilateralis is a fungus evolved for the cool, damp, tropical understory. Its optimal growth range is a cool 20 to 30 degrees Celsius.
Okay.
Crucially, its growth and metabolic activity cease sharply above 30 degrees Celsius. To infect a human, its fundamental enzymes and proteins would need to survive and function at a temperature at least 7 degrees Celsius higher than its absolute maximum.
That's not just a small adaptation. That's a fundamental physiological overhaul. It has to rebuild its whole engine.
It would require a massive fundamental evolutionary leap. For a fungus to function at 37 degrees C, it would need to completely restructure its entire proteome, the entire set of proteins it uses for everything from energy production to cellular division.
So it's a good comparison.
We'll compare it to a related entomopathogen like Bovaria bassiana, which has an upper thermal limit of about 33 degrees C. The challenge is still huge. Now contrast that with the successful human pathogen like Candida auris, which we will discuss next. C. auris not only thrives at 37 degrees C, but can survive heat shock up to 42 degrees C. The gap for Ophiocordyceps is statistically improbable over any short time frame.
And beyond temperature, there's just the sheer phylogenetic and physiological gulf between an insect and a mammal. Our defense systems are entirely different worlds.
The physiological distance from ants is huge. Humans possess robust adaptive immunity, T cells, B cells, antibodies, a complex targeted defense that takes weeks to activate, but which insects completely lack.
So a slow-growing fungus wouldn't stand a chance.
A slow-growing fungus like Ophiocordyceps, which takes 9 to 14 days just for incubation, would be easily detected, flagged, and systematically eliminated by the human immune system long before it could ever establish a systemic network or target muscle tissue.
And even if, by some miracle, it got past our immune system, the neural incompatibility hurdle seems insurmountable. The chemical keys that unlock the ant's motor system would simply not fit our locks.
They wouldn't. The chemical signals it uses to manipulate insect-specific neurotransmitter ratios, for instance, octopamine, which plays a major role in insect movement and stress response, would not fit the locks of human neuromuscular junctions, which rely on acetylcholine.
It's the wrong language.
It's the wrong language for the wrong machine. Our neural architecture is centralized, insulated, and exponentially more complex. Controlling a bipedal, massive mammal with a shielded, centralized brain to perform complex, targeted behavior is simply biologically infeasible with the specialized insect biology the fungus has evolved to exploit.
So if we take the fictional scorecard and we score the fiction against the scientific reality we just established, the results are overwhelmingly in favor of reality.
Let's run through it.
Fictional concept 1, brain infection. The idea that the fungus takes over the mind.
Inaccurate. We know the fungus avoids the brain entirely and controls the host peripherally, at the muscular level.
Fictional concept 2, rapid turn or turning infected in minutes or hours.
Completely inaccurate. The real-world incubation period for Ophiocordyceps is a slow, methodical 4 to 14 days, building biomass stealthily.
Fictional concept 3, bite transmission, passing the infection through saliva or a bite.
Also inaccurate. Transmission in the real world is via airborne spores landing on the cuticle, not through body fluids or biting.
Okay, fictional concept 4, networked minds, a kind of fungal, telepathic hive mind.
That's an exaggeration. Fungi do form physical, coordinated networks inside a host and in the soil through their mycelium, but there is zero scientific evidence for telepathic coordination across hosts.
But the final concept, climate adaptation as the driver for fungal evolution, that one is the real nexus of fear and fact.
That is the one concept that aligns perfectly with urgent scientific reality. The premise that a warming world narrows the thermal barrier is scientifically sound, and it's happening right now.
So if the zombie ant fungus is fiction, but the mechanism of climate-driven evolution is fact, what is the real danger? We need to shift our focus now from the specific fiction to the real data-driven danger posed by fungal adaptation in the Anthropocene.
This brings us right back to what Dr. 𝐏𝐂𝐃𝐂𝐂's work has been warning about, the 𝐏𝐂𝐃𝐂𝐃 hypothesis in action. As the average ambient temperature of the planet continues to rise due to climate change, that precious 7-degree thermal gradient, the gap between the external environment and our internal 37°C body temperature, is narrowing.
It's shrinking.
It is. And this directly and continuously compromises the efficacy of our natural multi-million-year-old thermal barrier.
So fungi that survive increasingly hotter summers and are isolated from, say, urban heat islands are essentially getting a free pass to test out thermotolerance at temperatures closer and closer to ours.
Precisely. We are inadvertently applying a powerful selection pressure on the entire fungal kingdom, selecting for thermotolerant species. They're being trained by environmental heat to survive inside a mammal.
And we can see this happening.
Research already demonstrates this happening in real time. For example, studies in places like Baltimore have shown that environmental fungi isolated from urban heat islands, which retain heat due to concrete and lack of green space, are already significantly more thermotolerant than genetically similar strains isolated from cooler surrounding rural areas. Cities are now functioning as evolutionary crucibles.
And fungi are inherently masters of adaptability, which makes this threat particularly concerning.
They are notoriously genetically plastic. Fungi utilize robust mechanisms to quickly adapt under environmental stress. One of these is transposon movement, often referred to as jumping genes.
Jumping genes.
These are segments of DNA that can move around the genome, accelerating mutation rates and enabling rapid reorganization of genetic material under stress. Heat stress, specifically, can increase this transposon activity, accelerating the acquisition of vital traits like thermotolerance and, critically, multi-drug resistance at the same time. This potent combination makes them highly responsive to our climate pressure.
And if we are looking for evidence that this thermal barrier has already been breached, the most alarming proof is the emergence of a human pathogen we've only known for a little over a decade. Candida auris.
Candida auris is the stark, grim warning that this entire scenario is already unfolding. This pathogenic yeast was completely unknown to the medical community before 2009.
Completely unknown.
But the way it emerged is what truly terrified epidemiologists and mycologists worldwide. Genetic analysis of early isolates showed that C. auris did not spread from a single outbreak point. Instead, it emerged almost simultaneously and independently on 3 different continents: Asia, in India and Pakistan, Africa, in South Africa, and South America, in Venezuela.
That pattern just screams environmental origin, doesn't it?
It is utterly inconsistent with typical human-to-human pathogen spread. This pattern strongly suggests that a single global environmental shift, the rise in global temperatures, allowed a previously benign environmental fungus to cross the thermal barrier threshold in multiple places at once, becoming a human pathogen.
And now it's a crisis.
Now, C. auris is a major public health crisis in healthcare settings globally. It's often multi-drug resistant and causes high mortality in vulnerable patients. It serves as powerful real-world proof that the fictional premise of climate change driving fungal evolution is already scientifically realized.
So the real Last of Us scenario isn't a mind-controlled monster, but a slower, more insidious threat emerging from the environment that our defenses are not optimized to handle. What are the projected real-world mycoses we should be paying attention to as the climate warms?
We face 3 key dangers. First, we will see increased fungal asthma and allergies due to greater spore loads in the atmosphere from longer, warmer growing seasons and altered air quality.
Okay, that makes sense.
Second, we face critical food insecurity. Fungal blights, such as the highly destructive wheat rust, will thrive in warmer, more humid conditions, destroying staple crops globally and leading to massive agricultural and economic crises.
And third, the most direct health threat:
the emergence of new mycoses. We will see the geographic range of established environmental fungi like coccidioids, which causes valley fever, or histoplasma, which causes histoplasmosis, expand dramatically northward and poleward. These fungi are adapting to heat, they are expanding their infectious zones, and that increases the frequency of human contact and cross-species jumping. The danger is not external takeover, but internal compromise driven by relentless, heat-driven adaptation.
This has been a true deep dive, moving from fiction to complex biological fact. We started with a monster, but we ended up with an evolutionary marvel. Ophiocordyceps unilateralis, a fungus that is an ancient, specialized neuromuscular puppeteer, not a brain invader.
Exactly.
We learned that the fungus is kept fiercely in check by ecological constraints, like the hyperparasites that save ant populations, and that the leap to functional control of humans is biologically insurmountable due to our robust, complex thermal and adaptive barriers.
The core takeaway, however, remains profoundly urgent. While the threat of the specific zombie ant fungus is fiction, the underlying central mechanism that climate change is actively driving fungal evolution by narrowing our thermal defenses is a valid and pressing scientific concern happening right now. We are inadvertently training our microbial enemies to survive inside us.
So what does this all mean for you? The real danger is not the monster that hijacks the nervous system, but the gradual, heat-driven evolution that is dismantling the thermal shields that have protected mammals for millions of years. This process is actively blurring the line between environmental fungi and human pathogens.
This is a slow burn.
It is. And when you gain knowledge of an ancient host-parasite war, you gain valuable insight into how knowledge of those conflicts can inform our defense against a contemporary invisible enemy. We have to recognize that the most successful threats adapt subtly and persistently, not violently and chaotically.
Absolutely. The next global health crisis might not be a fast-spreading viral chaos, but a slow, insidious, heat-trained fungus.
Food for thought indeed. Thank you for joining us for the deep dive. We'll see you next time.